So yeah, now we're ready for Alejandro to actually demonstrate submitting a test data set. And Alejandro, are you going to do like what kind of data set or data are you going to be demonstrating?
I'm going to be demonstrating a data set. It's a mouse data set with, one of the most typical cases, like some micro CT data and some RNA-seq data. Probably that wouldn't be that wouldn't necessarily completely match Marie's case if she's interested in submitting human subject data. But I think I can try to go forward with that case and try to go fast the parts that are probably not that important for her, and then talk a little bit more about how will be the specific case if she submits subject, human subject data. But
I think, Alejandro, I will be okay with that. It's pretty straightforward, even if these are mice data and not human data. Don't worry about that.
Yeah. Plus, it's we don't know exactly. It's very likely that you might end up using some of these, even if it's human, because maybe some of that human data may be shareable if it's fully de-identified, or sometimes we have cases where, for example, when they upload genomics data, some part of the genomics data is never really identifiable, like the big wheat tracks and things like that. And that part of that data sometimes gets uploaded to the site in the way that I'm going to present now. So I think it will still be useful, but I can try to make some of those parts not too long, and then we can talk more.
Thank you.
Sure. So let me share my screen. I think that most of these things are going to apply. Can you see my screen?
Yes.
Yes.
So let me start with a little slide that talks about kind of the process. This repeats a little bit some of the things that Rob already mentioned. So basically the process from the moment you submit your data submission request until you start creating the new data set can basically be outlined like this. So first you go there, you submit the data submission form that Rob showed. You enter all that information. We get that form. It goes through a process of sort of vetting and saying whether the FaceBase want that data or not. And once that decided we want to get the data, you will receive a link. We will basically create a group that is used for authenticating into our system, into our catalog for you and people in your group. I mean, we have a pretty sophisticated way of securing our data, and the way you authenticate so that you can only see and you can only see and only modify the data that belongs to you, and nobody else can modify, edit, or see your data if it's not yet released or while you're working on it. So in order to do that, we do that through a system that is called Globus that provides us the authentication rights. This has nothing to do with the Globus transfer data or anything like that. It doesn't mean that we use Globus to move our data or anything like that. We're just using their authentication mechanisms. So once we go through that, we create your project with the information that you provided in the data submission form, and we make you a part of this group. And once you do that, then you can go to your project. You need to log in first so that you can see your project, because since initially hasn't been released yet, so only people that is in that authentication group can see it. And then you go through the, once you get in your project, you go to the dataset section and you start creating your data sets. So let me start by demonstrating some of that. So let me go back to now. Is this big enough, or should I try to make it a little bit larger?
No, it's
Yeah, that's
okay.
better.
It's okay.
Okay, so we'll start typically at the FaceBase, front page as usual. And typically when you will start creating data set, the entry point is basically your project. So like I said, once you submit a request to submit data, we create this project for you, which is sort of like at the level of organization of the grant. Some scientists will have several grants and they don't want all these data sets to be put in the same bag, so they will have different projects. Let's say you will have a project for human data with NIH, and then you have another project with mouse data from another organization. That would probably be put into different projects. So you will start your exercise by going to your projects, and the way you do that is you go into our data browser section, you go to the projects, and you will typically look for your project. So let's say I will make all this demonstration in a project that I know that is called the demo project, the FaceBase demo project, and I see the FaceBase 3 coordinating center, but I don't really see that project. And the reason I don't see that project is because I'm not logged in yet. So it's not a public project. And since I'm not logged in as a user that is included in that project, I will not see it. So I will click login and log in using the authentication user or user that is being already added to that project. So there are two ways in which you can do that. You can do it through your organization. So you can go here in your first part and look up for your organization, which is basically the name of the university or school. Most likely all of the organizations are listed in here, and you can, if you have it, you can use your school authentication mechanism to get connected and be granted access. Or the other mechanism is using directly a Globus ID. For most of these exercises, I use a Globus ID, so I will click in here saying Globus ID sign in, and then click one of the users that I have. And you will see now I can see the FaceBase demo project because as that user, I have been already added to that group. So I have permission. That means that I have permission to go and see this project, even has not been released yet. So here I will see all the information about my project, basically an abstract of your research, the PIs. Let's say here I listed Yang Chai and Carl Kesselman, other project members. You can add additional people from your group in there. And all these things, once you are logged in here, you can actually edit them yourself. So we will create sort of like initial skeleton, but then you can go and do any changes you want. So I will start them by navigating down here to the dataset section, and we'll start creating a new dataset by saying add records. I don't want to say, I want to mention here that we have this other section that says QC issues, that once you start creating data sets, we have sort of like a monitoring system in the background that checks for inconsistencies. Like if you have created an experiment, but that experiment doesn't have some of the necessary parts, the system will kind of identify that, and we'll create that QC issue here. So you will see them listed as things that you probably had to work on for the data sets to be released. So I will start creating a new data set here. I will click Add Records. And here you basically get to the initial or basic part of the data set. So you start a title, let's say that is, as a publication, something that describes your research and what the data is about. I will put something like micro CT and RNA seq mouse data of E13.5 mouse, something like that. Hopefully more descriptive than that. Then you will have a description that we would like this to be as rich as possible in terms of, someone that looks at this and can get an idea of what type of experiments you conducted, the characteristics of your biosamples, and something that can be indicative of what is contained in this data set. So, image and sequence, something like that. Then we have a section for the study design, which is it's probably sometimes it has some sort of overlap with the protocols, but sometimes they can be like summarized versions of protocols, or something that is a little bit more technical than the description. We use, blah, blah, blah. And then what I wanted to mention is that in these fields that have these icons in here, that means that they are basically a markdown text, or basically rich text. So it's you can add images, links, bullets. So a little bit more editing than capabilities and plain text. And then the next two fields are whether it's human subject or not. For example, in this case, if the data that you are submitting contains some section of protected data, they will probably select this, switch this to yes, and then this field whether you will want your data set to be assigned a DOI or not. Most of the time we say yes. There's no reason why you say no, because that's typically the unique identifier for your data set. So once you completed that sort of initial part of the data set, you click save, and you basically have started your data set. So right now you have a very basic description of your data set, and now you will see that there's a lot of additional fields and things that you can enrich and add to sort of explain what your data is about, and tag the data. Before going into that, I want to show you something, is that on our user interface, in the top right here, we have this button called Hide Empty Sections. And by toggling this button, you basically make disappear all the things that are empty, that you haven't been added any information yet. And that is useful as we go along to see what things you have already completed and what things you might want to add to. So just want to show you that. Okay, so once we see that here, what we see here on the top are what we call the high-level tags of the data set, and also some additional information like, the contributors, the publications, the protocols, related data sets, protected human subject with yes or no, and the data use limitation. So these are sort of additional information for the data set. they're not required fields, but you can add to them. And some of these fields will be actually populated automatically for you after the data set has been submitted. We have nightly processes that run and fill in this information if you haven't added them. So for example, the contributor field, everybody that is in your project will be added as a contributor for this data set. But if you have someone that is not listed in your project, let's say it's not a PI and it's not let's say a postdoc in your group, you can go and add that person, that additional person. So you will click link records and select one of the users that are already in our system, or you can add a new user. I just want to mention that. Let's say I'm going to add myself. Then, for example, if the next field is the publications, if this data belongs to a paper or a number of publications that you have, then you basically click add record, and the way you enter that is by enter the PubMed ID of the publication. Click save, and then you go back to the main page of the data set, and you will see now this. Sometimes you need to refresh the PubMed ID entered in the publication. Something I want to mention is sometimes that some of these fields, when you click to add record, it will create a new tab, depending on, that also depends in the browser sometimes. But anyway, so, if for some reason you end up, after you enter the field, you end up in this new tab, you can always go back to your previous tab and continue working on your data set. The next row, the next lines are the protocols. And as you can see here, we don't have a way of entering a protocol here at this point. And that's because this protocol section will be populated automatically once we enter the protocols into the experiments. Then we come to the related dataset section. That means that if you have the data that you enter is related to some other data, let's say in some other database or some other repository like dbGaP or Geo, or you have FaceBase data sets that are somehow interconnected, you can create those relationships by just entering the ID of the FaceBase data set or by the URL of the dbGaP or Geo or any other repository where you may have related data. Okay, so the next ones are a list of tags that basically are these are basically most of the information that the system then uses to characterize and tag and search for the data. So all the tags that we talked about initially uses the information that is entered here. So for example, I know that I'm going to have microCT and RNA-seq data. So under experiment type, I will click link records and then search for that. And then I can also search for RNA-seq and find the RNA-seq assay. And now click link. And now we have, I basically tag my data set with two types of experiment, two experiment types tags. Same things for species. And I can select multiple. I mean, let's say I have this data set, I happen to have human, mouse data, I can add more than one. Many of these tags, even if you don't fill them up at this point, they will get automatically populated based on the description from your biosamples or your experiments in a nightly process. But you can here add additional ones. So sometimes there are sort of like synonym terms and that may want to add that, you know, that are helpful for people searching the data, and you can enhance that, add this additional metadata to it by adding new terms, but if not, they're going to be populated from the experiments or biosamples. So this is sort of the first part of entering the data in the in the data set, and then we go into what we call sort of the organization of the structure of the data set. Let me switch very rapidly to a slide so that I want to show more or less how I think in organizing this data before creating the data set. Oops. So, like Rob sort of mentioned, actually, let me go back here. If we go back to our documentation page, you know, I think Rob already showed this picture before. So this is the main sort of way in which we organize our data. We basically have the data sets. The data set have experiments. The data have experiments and biosamples, and then the experiments and the biosamples are sort of tied together by what we call a replicate. And then we add the actual data files or assets to the replicate. So this was something that sometimes can be a little bit confusing. This is something that we sort of artificially introduce in order so that we can reuse biosamples in many experiments. But sometimes, really, most of the time there can be a one-to-one relationship between a replicate and a biosample. So this is the main idea. Now I'm going to show you how I sort of try to organize it for this case, and how I'm going to go sort of hierarchically from the top to the bottom in creating this entity. So I have already created this data set. I already added some basic information about the data set. Then I will go and create these 3 experiments, right? Because I have one experiment for, let's say I know that in my data set I have some control data and I have some mutant data, and I did some microCT experiments and I did some RNA sequencing. So I will create one experiment to describe the microCT of the control biosample, another experiment to sort of gather the data of the microCT for the mutant, and then I will create another experiment to sort of associate the RNA sequencing data of the mutant. And then I will create these 2 biosamples. One is the E13.5 control biosample, and the other one is the E13.5 mutant. So this typically try to correspond to the physical entities that you actually perform an experiment on. And then I will create this, what we call replicates, that basically connects the biosample and the experiment. So you will see that each replicate is associated to one biosample, to one experiment and one biosample. So another replicate connected to this experiment and this biosample, one more replicate that connects the mutant biosample to the RNA experiment. And then once I create that, I will add the files to the replicate. And that's pretty much the how the projects are organized. Then at the, like we described, for each experiment, we want to associate a protocol. So we'll add the protocols to the 3 experiments. Actually, I'm missing here an arrow, but I will also associate this microCT protocol to this other microCT experiment. And then that's pretty much, I'm done when I've done that. So let me go back to the data set that I was working on. Okay, I had to go back to data sets and go back here. So I'm back at where I left off before I started creating the experiments and the biosamples. And again, you see, I will look in here to get rid of all the clutter, see what I have already done, which basically the only thing I've entered is some basic information on the data set. So now I will go down to the biosamples and experiments section and start creating those experiments. So I will click Add Record. The first field is the experiment type. So this is going to be my microCT experiment. I search for microCT experiment type. Again, these are all terms that we have taken from some known ontologies. So it are terms that they're not, and you cannot enter any term. They're terms that come from control vocabularies. And we maintain those, and if there's some that you need that for some reason is not there, because no one has requested it yet, then you can send us an email and we add those terms to our vocabulary. Then I will give a local identifier that, it can be any kind of tagging, or we prefer people to enter if your lab has some sort of scheme for identifying experiments. We'll like you to keep that. If not, I will give some, descriptive, like microCT E13.5 control. All these other tags are not really pertaining to this type of experiment. They're more for sequencing or ChIP-seq experiments, so I will leave them as they are. So now I'll go to try to create, I said I was going to create 3 experiments. So one thing that I want to show about our interface, which is very handy when you are creating many of these, is we have this feature of creating clones of whatever you have already created. So I can add 2 here and press clone, and basically I create 3 replicas of what I already entered, and now I can do some modifications and then save them all at the same time. So the second experiment is also going to be a microCT, but instead of being for the control, it will be for the mutant. So I will change that to mutant. And for the third one is going to be the experiment for my RNA sequencing. So I will change the experiment type. I will search for RNA-seq. I will select RNA-seq assay, and it will be for the mutant. So let's say I only did the RNA-seq of the mutant and not of the control. If I would, I would have probably added one more experiment to make it clear. So once I've done that, I click Save.
I think it's the local identifier for the RNA-seq assay. The local identifier still says microCT, which is the same as the previous one. So you see in the third record. local identifier.
Okay. So
Yeah.
Okay.
But that's a
Okay.
good point. It's like it won't let you save the same
That's good. Exactly.
local
Exactly.
identifier.
Yeah. Yeah. Thanks for that. Was a good video. Okay. So yes. So this is an RNA. So they need to be unique at the experiment type and ID. Okay. Thanks. Okay. So now I have created these three, and then I navigate back to my previous panel, and once it refreshes, I see that I created these three experiments. So the next thing, right? So if I go back now to my slide, so I basically went through this row now. I keep going down, and I'm going to create the two biosamples, the control and the mutant. So I click add record on the biosample. Again, I will use we know that, a lot of the labs, they have very well developed schemes for identifying the labs, their biosamples and experiments. So I would like them to use that if they are available. If not, I will just put E13.5 control, and then start selecting the metadata. This is a mouse sample. Specimen, let's say, is a tissue specimen, but you can select other ones. You can, if you are targeting a specific gene, or for genotype here, I will put wild type, because it's the control. Stage, I will select E13.5. Let's say anatomy, let's say this is the mandibular process. I know that's a process. Let's see. And as you can see, we have a very large number of anatomical terms here, and we basically have all terms from the Uberon ontology. So whenever possible, some terms are not in the Uberon, so we have them as internal terms. But whenever possible, like users to use the term that comes from Uberon. So in this case, I would probably pick the mandibular process mesenchyme, let's say, from Uberon. And this is just an example. Then we can enter the liter, let's say, if you have some information about that, and the collection date. Let's assume it was collected today. So here now I'm done creating my first control biosample. Then I will click clone again here to create a copy and change whatever things I need to change. I will change the identifier to be called the mutant. Then the genotype is going to change in this case. Let's say I will use LHX6. I think it's a good example for a genotype. And all the rest, let's say they come from different leader. I'm going to change that. And then we'll click save. And I will navigate back to my previous tab. And now I have created the two biosamples. So now the next step is creating these replicates that basically tie together the experiment to the biosamples that then I will use to add the data. So I will go back here. I will go basically experiment by experiment. I navigate into the experiment. I go to the section that says replicate. add record. And here, basically, what I need to do is select the biosample. And once I select the biosample, I will only see listed there the biosamples for this data set. So it's already kind of narrow. So this was the control biosample. So I select that one. In many cases, we do have several replicates for each experiment. So you will select additional biosamples and then increase the bioreplicate number sequentially 1, 2, 3 to show that there are basically samples that belong to a true replicate group. So once I created a replicate, I have different ways of navigating back to where I was, because since I created a new tab, I can basically go back here and see now that this replicate's been created. And then to go back one level up again, what I do is typically I use the links here as a navigation. So now I'm in this experiment. I created a replicate. I want to go create a replicate for the other experiment. So what I will do is I will go back to the click. I will try to go back to the data set by following this link, basically. So I click in there, navigate back to the experiment part, and do the same with the next experiment. But something to notice is that since now I have created this replicate that connects the biosample with the experiment, I see listed here all the characteristics of all those biosamples. So that gives us additional information is presented in this view. So we know the replicates, we see the species, the stage, the anatomy, the genotype. That's information that comes from the biosample. So I will do that again. Create one replicate now for the mutant biosample. Navigate back to the experiment, navigate back to the data set. I can use this menu items on the left if I want to go faster. So I can go back again to the experiment section and do it one more for the last, to add the last replicate. I only did RNA sequencing on the mutant, so I will only select the mutant here. Go back to the experiment, navigate back to the data set, and now again I will use these hide empty sections to look at what I've done so far. So I created, so basically now I have pretty much a structure of everything I wanted to do. So I have the data set, initial tags, the experiments, the biosample. So before going to the step to add the data, what I will do is associate the protocols to the experiments. So to do that, I will go again experiment by experiment. I will navigate down to the experiment, go in the protocol section, create link a record. And like Vu mentioned, you can, if you have already created a protocol, you can just search for it. Let's say I will use one of the microCT protocol from Child Lab. So I will select that one. If I will not have found any protocol or it's a new protocol, then I can create new and go through the process that Vu outlined. And you can select more than one protocol here too. But in this case, I will select only one, click link. I will go back to the data set following this link and repeat the process for the other experiment. add a protocol. And then do that to the last one. In this case, it's an RNA-seq experiment, so I will add one of the existing RNA-seq protocols, and done. So basically, at this point, you see I already see listed the protocols here in the dataset section. So I will toggle this again and basically see what's already filling. So if I go back to my sort of schematics, I have basically done everything here except for adding the data files. And that's the next step.